Here's a little video to kind of illustrate it. This is actually how it kind of looks, except there's no labels on the actual.
Translation is the process by which the information contained in messenger RNA is used to direct the synthesis of a polypeptide. This information is carried in the sequence of bases on the messenger RNA molecule. The polypeptide can be assembled once RNA binds to a ribosome. Ribosomes consist of large and small subunits. The large subunit has two binding sites for transfer RNA, the P and A sites. Transfer RNA carries an amino acid, which will be incorporated into the polypeptide. Its anticodon is a triplet complementary to the codon on messenger RNA that specifies that particular amino acid. A ribosome assembles on the start codon AUG. Transfer RNA with the anticodon UAC and carrying the amino acid methionine binds to the codon. The transfer RNA is in the P site of the ribosome. The A site is available for a second transfer RNA with an anticodon complementary to the second messenger RNA triplet. The amino acid carried by the second transfer RNA binds to the methionine. The first transfer RNA leaves the P site, and the second transfer RNA moves there, still bound to messenger RNA. This brings the third messenger RNA codon to the now empty A site, and the appropriate transfer RNA can bind to it. The third amino acid is added to the chain, and translocation occurs once again. This process of polypeptide chain elongation continues until a stop codon is reached. A release factor binds to the A site. It carries no amino acid, but facilitates release of the polypeptide and the messenger RNA from the ribosome.
Isn't evolution wonderful? And that's the simple version. Very, very simple version. It's much more complicated than that. Proteins. Proteins are functional units of the cell. Very specific sequential orders and three-dimensional structures are dependent upon the proper sequential order of the DNA upon which they're based. Some flexibility is there, depending on the location and class of amino acid letters, can change in protein sequence without the protein-based system losing complete functionality. So there's a little bit of flexibility. Like, for example, if I changed one letter in a paragraph of a book, would the paragraph still be readable? Most likely. You probably could have changed like 3 or 4 letters, probably still be readable, right? Most likely you could. But after a certain point, you change enough letters, it no longer makes any sense, right? Same thing happens with proteins. They can sustain a certain degree of flexibility, but after a certain point, they can no longer change farther without a complete loss of that type of function in question. In other words, there's a minimum specificity requirement for protein function of a certain type. Same thing for English language. Okay? So let's talk a little bit about Mendelian evolution, or changes that aren't Mendelian variation is what I like to call it. It's interesting to note that most of what Darwin observed was Mendelian variation, not actual Darwinian evolution, as far as the basis, which is kind of ironic. For example, finches can get large beaks, small little beaks, all these things within like 2 or 3 generations. And then depending on the change in the environment, and if the environment switches back, these little beaks can switch back and forth, just very rapidly depending on the environment, sometimes within a couple generations. All these changes can happen, which Darwin thought would take long periods of time and random mutations. But these changes are not based on random mutations at all. They're based on Mendelian variation, which is distinctly different. Same thing with all these breeds of dogs. These breeds of dogs, by and large, there's some differences that are based on real mutations. But by and large, breeds of dogs are based on Mendelian variation, not random mutations. Okay, same thing with the peppered moths. Here are the famous peppered moths and all these college textbooks. And this is an illustration of evolution in action. There's no such thing. These differences in color of the peppered moths are Mendelian variation. Okay, same thing with the pea plants for Gregor Mendel. Gregor Mendel, if he had become famous before Darwin would have had a much harder time making his theory popular. Gregor Mendel is the father of genetics. He studied pea plant hybrids and discovered the principles of basic genetic recombination. Darwin was not aware of Mendel's work, even though they lived at the same time. So, in a nutshell, genetic recombination, when the chromosomes come together in meiosis, they kind of swap information with each other, but they do so at very specific points that are predetermined. In other words, no information that was originally present in the gene pool is actually altered. It's just given a differential expression. Okay, for example, a gene is a stretch of functional DNA that often codes for a protein. Genes occupy specific positions called loci. For a given locus, different versions of the same gene or alleles will fit or work in this position. They're called different types of alleles. Just like different types of wheels will work in the same wheel location on a car. Can you trade out wheels on your car and put different kind? You go to the car body shop and you say, I like that rim and that kind of wheel and on my car. And somebody else would say, no, I like the other rim and the other kind of wheel on the car. And the body shop, let's say, has 15 different rim options and 20 different wheel or options, and you can mix and match all those options. But are there any other options besides those options that you can do for the wheel at the body shop? No, there's only those options available. Those are the alleles. And also, can you take the wheel and put it on the wheel for the car and put it in replace of the motor and have the car still work? No, the wheel has to go in the wheel spot on the car. Makes sense? For the car to still work. Same thing with Mendelian variation. You have all these different allele options in the gene pool, but the alleles must go in the right spot for them to work properly. Make sense? What's interesting about sexually reproducing organisms is that we have two copies, two chromosomes for every trait. And so these two copies can mix and match each other. And so which option for which chromosome actually gets expressed? Well, that depends on which one is dominant. There's this term called dominance and recessiveness. And the dominance, and there's also codominance. But to be simple here, let's just talk about dominance and recessiveness. The dominant one gets expressed and the recessive one does not get expressed. And so depending on which alleles are put there together or not together, you can have all these different options. You can have differences in flower color, purple or white for peas, or you can have long or short stem length, etc. All these different options, just like you're on your different types of wheels on your car or different types of colors on your car or whatever. You can have different options, but the gene pool of options itself, the resource of options stays the same. Okay? We already talked about dominance and recessiveness. So how do you express a recessive gene? Well, here you got a recessive, a green allele and a yellow allele, and the expression here in the first generation is a mixture of Y's and G's, of yellows and greens. So up here you can see the phenotypic expression, what will it actually look like. It will only look yellow even though it has genes for green. See this? Yellow even though it has genes for green. So which gene is recessive? Green is recessive because it only expresses yellow, even though it has the gene for green. So how do you express green? You have to have two genes, two green genes, two green alleles for the thing to be green, right? All these have two greens, and so those are green. Otherwise, if it has even one yellow, it's going to be yellow. And then you can have multiple alleles. Some traits require many alleles to enhance them, like size. If you have more big-sized genes, you're going to have a bigger dog, for example. And if you have fewer genes for size, you're going to have a smaller dog. But there's a limit to how many size genes can be expressed by a given organism. That's why you can't get much bigger than a Great Dane in current doggy gene pool. And you can't get much smaller than a toy Chihuahua for smallness. You can't get can you breed down to a thimble-sized dog? No matter how hard you do your selective breeding? Yeah, cute. It would be cute, but you can't do it. And can you breed a dog the size of this room? No, you can't do that. Okay? So there's limits to how Mendelian variation will work. So this difference expression of a static gene pool. And so what does it take to evolve a different gene pool? For example, using Mendelian variation alone, can you turn a cat into a dog? Or a lizard into a bird? No, because the gene pool is static. Using Mendelian generation alone, it only has a limited number of options. You can't add more options using Mendelian genetics. So how do you get more options? You get more options or more types of alleles using random mutations that actually change the number of alleles or the type of allele or the quality of alleles. Okay? So what are the odds? What are the odds that you're going to get alleles with certain levels of functional complexity based on statistics? Well, I like to imagine statistics based on a checkerboard of two-dimensional sequence space. How many different three-letter options are there in the English language system? You have 26 letters, right, in our alphabet. How many different three-letter sequences can you make with 26 letters? 17,576. So that's 26 to the power of 3. Okay, you have your number of options and you raise it to the power of the length of your sequence. Everybody understand that? That's how many options you have. So that is your total space of options. That encompasses every possible three-letter sequence you could think of. Understand?
Yeah.
Okay. So what are the odds that I'm going to start on a beneficial three-letter sequence and take a step at random and end up on another beneficial three-letter sequence?
Pretty rare.
Actually, they're pretty good.
I guess.
Okay? Here's the stepping stones. Here's the little checkerboard. Here's this little island. This represents three-letter sequence space, and as you go on higher and higher letters, the targets get rarer and rarer, relatively speaking, and I'll explain how that works a little bit. It's like stepping stones. If you land into the water, does natural selection work anymore? If you're swimming in the water, you have to swim randomly because nature only selects for what is increased in beneficial function, right? Does nature recognize the difference between Quisly-Gook and Quisly-Guck? They both have the same non-functional function, and so nature just flips a coin and says, "I'll take Quisly-Guck." So in order to make nature say a beneficial selection, a preferential selection, the mutation has to produce a sequence that produces a phenotypic functional change that is also beneficial. Okay? So at I don't know if I have the statistics here yet. At three-letter words, the ratio is about 1 in 18. The odds of taking a random step that you'll end up on another beneficial target, if you look up in the Scrabble dictionary, you can figure out the ratio. The ratio is 1 in 18. For two-letter sequences, it's 1 in 7. What do you think it is for seven-letter sequences? Like you kind of double it. Do you think the odds double? Or the odds decrease by double? No. The odds go down to 1 in 250,000. Okay, now let's go up to 28-letter sequences. What are the odds now? So it's 1 in 63 billion. Okay, so it's an exponential decline in the ratio. Yes?
Sorry, real quick. Talk about like beneficial to the person evolving. Is that like divine evolution?
No, I'm talking about if the mutation produces something where you can either make more kids or your kids survive better than everybody else's kids, then that's beneficial, according to theory of evolution. And it works. That works. But you actually have to land on a sequence that does that, and landing on a beneficial sequence depends on the odds of sequence space. And at low-level sequence space, three-letter words are interconnected, like I showed you before. Cat, hat, bad, big, dog, right? They're interconnected, and there's only a few little islands of water around that are not beneficial. The ratio is pretty high. For two-letter sequences, it's 1 in 7, right? But then it's an exponential decline. The water or the non-beneficial sequences in sequence space increase exponentially faster than the increase in beneficial sequences as you move up with higher structural or higher sequence and specificity requirements. So as you go up to like 7-letter words, all of a sudden you get a lot more water and a lot less islands that nature can select for. And the islands kind of get stretched out like gum, and they're kind of sticky, and they have these little bridges between them statistically. But then as you go higher, then the islands start breaking apart and the bridges start breaking between them. And then you keep going higher and the ocean just takes over and these islands become remotely isolated in sequence space. So then how to get from one island to the next in a remotely isolated sequence space? What is there? Does natural selection work to help guide the process along? No, it's purely random walk. So to get from this island to this island, the linear path is increasing linearly, or the distance between islands, as the linear distance increases linearly, or the direct distance increases linearly, the random walk distance increases exponentially. Okay, so because you have to, this is an average random walk I generated on the computer. Here's the island here, missed it, kind of close. Missed it, but it took a long walk. If you stretch the red line out, how much longer will the red line be compared to the yellow line? Exponentially longer as you increase the distance of the yellow line.